Okay, let's just start with the polymerase chain reaction. So this one is the polymerase chain reaction. So why we need the polymerase chain reaction? So first time this was discovered by the Carey and in polymerase chain reaction, the thing was to amplify a large quantity.
सर वॉइस नहीं आ रही है।
So your mic is off. You're not audible.
Sorry. Uh, okay, we start from beginning. Actually, there are some noise. Now this one is a clear, na?
यस सर।
Yes, sir.
Yes,
Yes.
yes, sir.
Okay. This we start with the polymerase chain reaction. So it's about the DNA sequences and their DNTP structure, which can extend the DNA chain, and then polymerase enzyme, which can use for the extension of that DNA chain. And then there are two big issues for the polymerase chain reaction. There are a lot of other sequences in the genome that we are not interested in detecting. Actually, they are specified sequences, and the amount of DNA in sample we are interested in is very small amount, so we can amplify a small quantity of DNA into a microgram quantity of DNA. With the help of this amplification, that will be a polymerase chain reaction. So we already know about that. This reaction was first developed by Carey in 1975. So these are the two big issues where why we need to amplify the gene sequences. So this is a genome, and to have to amplify that genome into a specific DNTP structure. The next thing is that how do we identify and detect a specific sequences in a genome? So there are lots of organisms by which we have to amplify that particular genome. And these are the list from some sequences. Suppose human is having the 3.4 billion base pair, whereas housefly having the 9 million base pair. So these are the HIV 9.7 thousand base pair. So these are the organisms which is having a different type of genome. So to maintain its integrity or stability or as a genome bank structure, we need a polymerase chain reaction to identify that particular genome. So the next problem for the to amplification that the human genome is 3.5 base pair. If the bases were written in strand about 10 point or 10 point scale on a tap measure, that will be a 500 366 miles. So this one is a large huge gap. Uh, and we can identify a 500 base pair sequence in a genome, and that will be the we can measure that genome uh, in a in approximate 3.4 billion species, which can measure in a somehow in a particular sequences. So this for that's why we need to amplify that particular genome into a large quantity. So by the amplification, how many molecules do we need to be able to see them? So for that amplification, suppose we isolate a genome in a agrogel, then we need to around to 10 nanogram of DNA for fluorescent stain. Then we go through a gel electrophoresis and then we identify a particular fragment of that DNA into different manner. So this is the data for the Avogadro number 6.02 or therefore 1.8 copies of that genome. So this one is a single gene which have a which also have a large quantity or in a large scale. So that's why in a single gene or a particular gene which can there is a need to amplify that particular gene into a number of copies gene. Next, how do we identify and detect a specific sequences in that genome? So there are two important objectives. First, there are a lot of other sequences in genome that are not interested in detecting, and the amount of DNA sample we are interested in is a very small. And both problem solved by the polymerase chain reaction. If there is a small number of genomes, so that can be amplified or that can be detected. And if the amount of the DNA is very small, so that can also be detected by this. So we can uh, we can solve this both these two issues with the help of the polymerase chain reaction. So how the PCR work? This one is the first thing. So there are certain needs which are used for the functional of PCR reaction. So they are need of distilled water, then buffer, one a DNA template strand, and a DNA polymerase enzyme, and a primer. One is a forward primer or reverse primer, and a DNTP. These are the four different types of nucleotide or DNTP for the assembly of the DNA chain, and particular a divalent cation such as magnesium ion, and a particular enzyme which can extend the DNA chain with its activity of 5' to 3' exonuclease activity. So this polymerase enzyme having the activity of 5' to 3' exonuclease activity, and all these requirement used for the PCR purpose. Next is suppose is a water, so this will be a charge-free water or N-free water. So this one is a media for all of the component and all of the buffer. So that's why distilled water will be required. And then the buffer, which can stabilize the DNA and which can also use for the polymerase reaction, and also used for the DNTP work. So that's why a phosphodiester bond will be formed between two DNTP or three DNTP, and buffer will be 500 millimole KCl, and it is a TIS SCl which is having the pH 8.3, and other buffer formation is Triton X100, which is also used for the buffer preparation. Next is a DNA template which contains a particular region which have to be amplified. Suppose if there is a gene of interest are very much small and you want to amplify that particular gene, uh, where uh, where sequences are unknown. So that's why you can use a unknown primer, in the sense that you have to use a satellite DNA molecule which can use to amplify the unknown base pair sequence. So the region which you want to amplify, or any desired DNA sequences uh, for which you have to amplify with the PCR, there is a purpose of to make a many copy of a particular gene with the help of the PCR and with the help of the cloning. There are two things. If you want to isolate a particular gene sequences, then you have to amplify. The better you have to use a cloning purpose. But in the sense of the PCR, it can be amplify a particular large amount of the DNA, or it can amplify a whole chromosome, or it can amplify a large amount of genome. So any desired DNA can be amplified with the PCR reaction. The purity of that DNA will not required, because any copy of the gene, if it is a genome of non-coding sequences or coding sequences, it doesn't matter. It can amplify all over DNA, and should be free of polymerase inhibitor enzyme, because there are primer activation, or there are a primer extension. So there will be enzyme which is having a polymerase free, or you can say polymerase inhibitor free enzyme. So that's why these are the need for the PCR reaction. Other is um, uh, except buffer and DNA template, there is a primer. These are the most important things which are required for the PCR reaction, that is specific for the end of amplified region. There are two types of primer, forward and reverse type of primer. Annealing templates should be known, and depends on the primer length. It will be the GC content or etc. If you add a alkaline phosphatase enzyme, and you can add anything on the, or oligo DT or oligo DG on the 3' end with the help of the enzyme terminal transferase. So suppose you add a oligo DG tail on the 3' end, so better you have to use a primer on the oligo DC primer on the 5' end. So with the adding of some known sequences or some known primer, you can amplify your desired DNA sequences. So as in cDNA library, you use a terminal transferase enzyme, then this terminal transferase enzyme actually find any oligo DC or oligo DG DNTP on the 3' end. So you know, if you want to extend this 3' to 5' chain, so better you have to use a primer which is having the complementary that of oligo DC sequence, or so you can use a oligo DG primer which can extend from 5' to 3', and it's about the length of primer 50 to 30, and the concentration, this one is a concentration for the primer will be used. So another thing is important, that is nucleotide. So there are 4 different types of nucleotide will be used, and this one is how a chain will be extended with the help of the DNTP. So there are 4 different types of DNTP which can use to extend the chain, and which can extend or which can join together with the help of the polymerase enzyme. So this DNTP is stored at the concentration of 10 millimole, and at the pH 7 it can amplify. And at least add 20 to 200 mm in a as a buffer. And why the magnesium ion concentration? So actually it is an essential cofactor of the DNA polymerase enzyme, and it is if there is a magnesium ion is not present, or the less free magnesium ion free medium, so there is the enzyme will not work. So for the it can stabilize the DNA double helix, and if there is a magnesium ion concentration too much, so DNA extra stable non-specific primer will bind, and then band smearing. So the magnesium ion concentration will be appropriate, and that will be 0.5 to 3 μm in the assay. Next is that DNA polymerase, and this one is the most important thing about the PCR reaction. So what type of the DNA polymerase will be required? Actually, the overall temperature for the machine, PCR machines, should be not higher than 55 degrees Celsius or 54 degrees Celsius. In some step, that will be higher up to 72 degrees Celsius to 94 degrees Celsius. So this the on average temperature of that machine will be on 54 degrees Celsius or 52 degrees Celsius. So there is a need of polymerase enzyme which can stable up to that temperature. So this one is a particular DNA polymerase enzyme which can isolate it from a thermophilic bacteria, Thermus aquaticus. So there is a DNA polymerase which is heat stable, that is block tag polymerase, and it can extend the DNA chain approximate 1 dNTP per reaction or 1 unit per reaction. So this is the DNA polymerase enzyme which can extend the DNA chain approximate 1 dNTP per reaction.
Sir, you are not audible.
Audible.
वॉइस नहीं आ रही.
Sir, your mic is off.
अपने आप ही ऑफ कैसे होना है? I don't know why it's automatically shut down. So now it is audible, na?
Yes,
Yes,
sir.
sir.
Yes, sir.
So this one is the application of a specific target sequences. PCR does not copy all of the DNA in the sample, and it copies only a very specific sequence of genetic code from a template DNA, and which is targeted by a PCR primer. And it does require the knowledge of some DNA sequences information which flank the fragment of the DNA which will be amplified or target DNA. So these are the primer which is attached on the 3' end, and that primer actually used to extend the DNA chain with the help of the polymerase enzyme. So this one is a basic a particular primer can attach on anywhere of the DNA template sequence. So, but it will be bind on the 3' end. So if there is a, you can use a known primer or unknown primer, and primer will bind on that complementary strand, and this complementary strand extend by the use of the DNA polymerase enzyme. So next is from there is a two synthetic nucleotide primers may be chemically synthesized, and each complementary to a stretch of DNA to the 3' side of the target DNA. One oligonucleotide for each of the two DNA strands, and that can be extended with the help of the polymerase enzyme. So here it is a 3' end, or one is a one strand, one complementary strand is a 5' to 3'. Another complementary strand is 3' to 5'. So primer bind on the 3' end. So this one is a 5' to 3' primer, and here is a 5' to 3' primer. And this primer will extend the DNA chain with the help of the DNA polymerase enzyme. So this one is a DNA primer or RNA primer. I am audible now.
Yes, sir, you are audible.
Okay. So why we don't use a RNA primer on the PCR reaction? Why we use a DNA primer on the PCR reaction? Why in vitro replication or in vivo replication we need a RNA primer? But instead of this, in PCR reaction we use a DNA primer. So why we use DNA primer in the PCR reaction? Because RNA is, anybody knows about that?
So RNA primer would be unstable. It won't
Yes,
be able to form a
yes.
complementary
Because RNA
sequence.
primer is too much fragile and it is unstable. So that's why we use a DNA primer in this. So This one is a reaction. Here is a PCR primer must bind to a sequence on either side of the target sequence on or in a opposite strand. So in a first view, there is a primer annealing site and here is a primer annealing site and region of DNA to be amplified by the PCR. Whereas in second view, when target DNA is single stranded, primer bind and allow DNA polymerase to work. So in this case, primer bind and allow to extend the DNA chain. So there are two steps by which a primer can amplify the DNA. And next is what is the purpose to use a primer and what is the purpose to use a two primer. Some PCR such as anchored PCR and nested PCR, we use only a single primer. So why we use a two primer? Uh, there are reverse primer and this one is a forward primer. So one strand actually one complementary or one template strand is 5 prime to 3 prime. That's why we need a primer which is extend the chain from the reverse backward. So that this one is a reverse primer and one chain will be extend from the forward. So this one is a forward primer. So that's why there are two primer will be used. One is reverse primer for the one strand and one is the forward primer for the second strand. And better we if we know about that particular sequence where the primer will be bind, so it will be better. So we can start from a beginning to the end. So we can extend the whole polymer whole DNA chain with the help of the known primer. And in a PCR reaction, the forward primer have the same sequence of forward DNA strand which will bind to the complementary reverse strand. And whereas then reverse primer which having the same sequence of the reverse DNA strand and bind to the complementary forward strand. So these are the two primer. Basically we need in PCR forward primer and reverse primer. Then setting up PCR reaction. So in a PCR reaction, the the requirement or basic requirement that will be the sterile water and 10x PCR buffer. One is a magnesium salt concentration and different types of DNTP and the primer forward primer and reverse primer, DNA polymerase, DNA template, and that will be the total volume of 50 ml because we have a up and down tube and up and down tube have a certain limitations. So that's why all the requirement of PCR reaction have a certain value. Next is a total volume which is used for the up and off tube. This is the table for that. You can note it. You can take a screenshot for that. It is a 1x and 20x component where we use a different type of sterile water, 10x PCR buffer, then magnesium chloride, then DNTP, two primers. The concentration of primer are DNA polymerase reaction and DNA template, and these are the total volume for the up and off tube. Then, or you can say PCR tube. So these are the some statics or the master mix for the extension of the DNA chain, and this one is a same thing. So, what will be the programming for the thermal cycler? So there are three programs for the thermal cycler. That is a denaturation process, and then primer annealing, and then extension of the primer. So initial denaturation will be occur on the 95 degrees Celsius for 3 minutes. DNA denaturation will be occur on the 95 degrees Celsius for 1 minute. Primer annealing for the 65 degrees Celsius for 1 minute. And it can be changed or it can be difference in a different protocol or in different books they are different. In some books they are written as a 72 degrees Celsius or primer annealing is a 54 degrees Celsius or 52 to 54 degrees Celsius. DNA denaturation is a 94 degrees Celsius for 2 minutes. So whatever it will be depend upon that particular genome or the complexity of the genome. The primer extension for 72 degrees Celsius for 1 minute. So these are the temperature and their time for extension of the PCR. and the PCR cycle. First thing about there are three steps for the PCR cycle. That will be the denaturation, which is 95 degrees Celsius, annealing of the primer for the 50 to 65 degrees Celsius, and extension of the DNA chain for up to 72 degrees Celsius. So, in the denaturation process, if you want to make a copy of that particular parental strand, so better you have to first separate it out all these strands with the denaturation process. So in denaturation process, all the both strands will be separated, and in vivo, this will be done by the helicase enzyme, which can unwind the helix, and both strands will be separated. So there is a space which is required for the primer to extend the DNA chain. So that space will be provided by the helicase in vivo, but in conditions of PCR, it will be provided with the help of the temperature. If you extend the DNA chain on the or extend the DNA buffer on the 95 degrees Celsius, the complete denaturation will be occurred. Both strands will be separated out. So, at what temperature the DNA denaturation will be started? Anybody know about that? What temperature DNA will be initiate to denature? I'm audible. Huh?
So you're audible, but we don't know the answer to the question.
DNA will be. I think DNA will be start from the 80 degrees Celsius.
The
For
95 degrees Celsius,
uh,
the
yeah,
95 degrees
that can depend for the complexity of the genome. If it is a prokaryotic genome, that will be different. If it is a eukaryotic genome, that will be different. Which one is a more complex genome?
that
Having the
is a,
5,000 base pair of gene or having the 20,000 base pair of DNA? Which one is a more complex genome? A genome having 5,000 base pair
The
or a
20,000.
genome having 20,000 base pair?
So with 20,000.
Why 20,000? The size of the genome make their complex, or the anything else which can make the complex, which can decide the complexity of that genome? Hmm? The complexity of the genome depend upon the size of the genome
So it
or
depends
anything
upon
else.
the number of the nitrogenous bases that are attached to the genome. So in 25,000, it would be more of the analysis have to be done.
Or there will be some non-coding sequence, intervening sequence, or coding sequence that can make the complex DNA. There is a quart value now. So the quart values depend upon the complexity of the genome. The DNA which is much more denatured in a certain time, that means DNA will be much more renatured at that particular time. So the renaturation step will be directly proportional to complexity of the DNA, or it will be inversely proportional to the complexity of the DNA.
the directly proportional, not directly proportional to its genome size.
Inversely proportional.
Yeah, yeah. So that is why it must be proportional.
Okay. So the double extended DNA template is denatured by heating typically to 95 degrees Celsius to separate the double extended DNA. So this one is the first denaturation process. Other is a annealing, which is from 50 to 65 degrees Celsius. The reaction is rapidly cooled to an annealing temperature, which allow the oligonucleotide primer to hybridize to the template strand. And about the extension of that primer, the reaction is heated to a temperature up to a 72 degrees Celsius, which can efficiently synthesize the DNA with the help of the thermostable DNA polymerase, that will be a Taq polymerase. So, by the three steps, denaturation, annealing, and extension of the primer, one cycle of DNA will be completed. So, whatever amount of DNA is required, that will be depend upon the how many cycles you have to run through. Next is the first is the denaturation step, by which the double-stranded DNA is denatured, and up to 95 degrees Celsius, the complete denaturation will be occurred. So both strands will be separated out, and there is a space to binding or annealing of the primer, by which the chain can be extended. And there is a one more thing: if you want to break, if you have to exceed the 95 degrees Celsius temperature, so that's why the breaking of the bond will occur. Breaking of the bond means it is the phosphodiester bond which will be break if you have to go through the above the 95 degrees Celsius. So DNTP will be break down, and all these strand will be break down. So better you have to the temperature will be between 94 degrees Celsius to up to 97 degrees Celsius. Temperature, if temperature is more than this 97 degrees Celsius, so that means the bond will be break down. The DNTP will be break down, phosphodiester bond will be break down. So that means the DNA will be completely denatured in the as in DNTP structure. So this one is the first denaturation process. Second is the annealing of the primer. The reaction is rapidly cooled to the primer annealing temperature, that will be 50 to 65 degrees Celsius, and which allow the oligonucleotide primer to hybridize to a single standard template. And on average, the annealing temperature will be 54 degrees Celsius, and that at that temperature primer will anneal only to the sequence that are completely to them on that target sequence, and the bond will be formed. So the primer and the template stand, the hydrogen bond will form, and that will make the chain to extend. So this one is step 2, and the temperature for the step 2 between 50 to 65 degrees Celsius, where the primer will anneal. One is a this one is in this case this one is a forward primer, and this one is a reverse primer. So there are two primer which can extend the DNA chain accordingly. Extension: the reaction is heated to a temperature depend upon the DNA polymerase used, and it is commonly a temperature of 72 degrees Celsius, by which the primer enzyme can extend or extension of the primer with the help of the DNA polymerase and some DNTP. So DNTP used to make the phosphodiester bond between them, and as complementary strand, DNA will extend out. This means that 72 degrees Celsius is the optimum temperature of DNA polymerase, and at this step the DNA polymerase synthesize a new DNA strand which will complement you to the DNA template, and that will be the 72 degrees Celsius for the extension of the DNA primer, which can extend the DNA chain. Now, and this DNA polymerase is a DNA dependent. This DNA polymerase is a DNA dependent DNA polymerase, like in vivo. It can extend the DNA enzyme. It can extend the DNA chain on the DNA template. So this will be a DNA dependent DNA polymerase. Uh, this one is a Polymerase reaction in which a time versus temperature that can represent the denaturation of the DNA strand, then annealing of the primer, and then extension of the primer. So at per cycle or number of cycle, the DNA can be denatured, and annealing of the primer, and then extension of the primer. So it is a gap between the cycle per cycle from denature to extension of the primer. This one is the cycle 1. Now, at the end of the PCR reaction, the specific sequences will be accumulated in a billions of copies. That will be amplicons, and in only 20 cycle, PCR can produce about a 2 million base copies of that target sequences. So this one is the extension. by which one cycle make the 2 copy and 2 cycle make the 4 copies at the power of at as formula 2 power of n. So by which we can make the different copies. So approximate several cycle we can make a microgram quantity of DNA from a few base pair of DNA. So this one is an example. If there is a mutation in any gene and you want to recover that mutation or find out that mutation, so you have to isolate that gene and then run a PCR primer, which can find out the sequence from any target region, and then design of the primer, which is using a primer designing tool. There are so many software which can design that primer. So this send which can use to amplify the template strand or particular desired sequence of that gene, and make sure that the area that you want to study is between the primer. So because primer can extend in the forward or reverse manner, and there are primer specific specific software that are blast or listed primer or prime time. There are many type of software which can be used to design the primer, and which can optimize your PCR and troubleshooting, and then you can use a particular gene, or you can find out the mutation in the genome or with the help of the PCR reaction or PCR prime. So the denaturation step is suppose in this case they are the gene sequence, and this one is a 5 prime to 3 prime stand, and this one is a 5 prime to 3 prime stand in this one is a forward stand and this one is a reverse stand. So there are sequence at the 3 prime end, which is shown is in white. So you can use a primer to attach on this white region on this 3 prime region, which can extend the DNA chain up to your desired fragment, which is on covered in a blue sequence. So you can extend your desired sequence, or you can extend your only gene sequence that will be need to amplify. So you can amplify that product with the help of the PCR. So first is a denaturation reaction, by which you have to first denature that particular sequence, then annealing of a primer. This one is a reverse primer. First is a reverse primer, 5 prime 3 prime. So it will bind on the particular known sequences, and which can amplify on the two denatured sequences. So this one is a forward primer and this one is a reverse primer. There are two primers which can amplify. This one is an extension. A Taq polymerase or DNA polymerase can extend the primer with the help of the 5 prime to 3 prime exonuclease activity, and that can extend the DNA chain. And this one is a Taq polymerase, which is a thermal stable polymerase, because up to 72 degrees Celsius, the polymerase should be stable, because at that temperature the DNTP will be make a phosphodiester bond and chain will extend. So up to that temperature, a Taq polymerase will be stable, and that will extend the desired DNA sequence. And then there are 1 or 2 or 3 cycles which can make to amplify the particular sequence. How can you analyze that the DNA sequence is amplifying or not, or PCR reaction get well amplified, or the primer have to extend the DNA chain, or primer is working efficiently? So there are some visualizing steps by which you can find out that primer is actually work or not. So after thermal cycling, tubes are taken out of the PCR primer, then it will be loaded onto the agar gel, and that it will be separated by the using an agar electrophoresis. So if there are certain band is available or visualized, so that means it can be extended by the PCR primer. So Here is an example by which you can visualize your particular product with the help of in the second case the band are not visible, but in the first case band are appear. So that means the strands are formed. Next is the visualizing result. The final result of the traditional PCR procedure is a gel with a series of bands. Bands can be compared against each other and to known to size standard, which can determine in the presence or absence of a specific amplification product. So that can will be visualized with the help of the axial electroforces. This is the original view for the gel that is visualized by the axial electroforces. Then uh, the use of the PCR in forensic DNA detection. It can also be used to identify the transgenic plant and in detection and quantification of the viral infection such as COVID or reverse. It is also used for the cloning purpose and it can also be utilized for the detection of ancient DNA as VNTR sequences, finding out the VNTR sequences, then amplify that VNTR sequences, and or you can make a RAPD with, and this RAPD can used to detect with the DNA fingerprinting to make any similarity between species. So it can also use to expression of genetic analysis. The PCR, the expression of any RNA or mRNA chain that can be amplified by the RT-PCR. You can express that analysis with the help of the PCR. So it is also used in a forensic science. So PCR have the ability to amplify even the smallest amount of DNA from a sample collected at a crime scene or any other scene, uh, which give the method to give the power when I use in criminal or in forensic science. The DNA from any type of sample, that is body fluid, hair or other tissue sample, which is amplified to create a nearly unique pattern of each individual, and this pattern can be compared to a suspect in any crime case. So uh, it was the first case in famous O.J. Simpson that will be solved by the PCR and which we publicized. And the genetic GMO food detection and GMO is actually it is a wide grown in the USA and other countries, and for various reasons, some countries require export to indicate the percentage of the GMO in their grain and any type of food shipment. So PCR can be used to detect the exact quantity of genetically modified food in a shipment by looking at the DNA that make up the food. So it also be utilized in that. And it is also in paternity testing. PCR powerful, very powerful technique to identify individual genetic makeup, which is made to invaluable for the use in paternity testing. And by the amplifying of the specific DNA fragment from a parent to the close relative, it is possible to reconstruct uh, phylogenetic analysis between individuals or phylogenetic similarities or any type of genetic disease that can be genetically inherited. So it can be recognized or it can be identified by the PCR. And PCR can not only identify relationship between people today, but it also used to identify the historical family relationship. In archaeology, PCR used for the many scientific studies in the field of the archaeology. It can reconstructing the Dead Sea Scrolls and it can identification of the paint pigments in cave paintings, and it can also determine the latentness between the individual in ancient Osiris, and it can constructing the fossil. It can be used to constructing the phylogenetic map with the help of the fossil's DNA or fossil samples. And then disease diagnosis. PCR is invaluable to in the modern class diagnosis, and that can be identified in disease-causing organism much earlier other than methods it took for the DNA of the organism and so not its protein or its effect or in our immune system. PCR has even been used to diagnose a disease of the past by amplifying minute amount of the disease related DNA in preserved specimen. So it can be helpful in disease diagnosis and in disease treatment. PCR not only be used in disease diagnosis but also in the aid of the disease treatment. Real-time PCR is used to directly monitor the amount of HIV virus in patients suffering from infection, and it can also use to monitor the virus persistent in the organism for a chronic as a chronic infection or as a acute infection. So that can be detect and the wildlife conservation. It can be identified in the it can differentiate between species and it is often used in the wildlife conservation research. PCR can be used to monitor trade in products from the endangered species. PCR can be used to monitor the ecosystem and the presence of certain species, and it also used to monitor and identify the individual animals. So the human project has identified tens of thousands of genes in the human genome, and it can be genes which are turned on or turned off, that can be also quantified with the help of the PCR. So it's all over the PCR. If you have any questions, you can ask. If anyone have any questions, you can ask.